Let's see. Is that good? You want to come up? Anyone else in the back you want to scooch closer? I'm going to show you some cool animals. Got stuff in a jar. You don't want to be close. All right, well, I'm going to get started, and thank you guys for coming. And like Kristen said, I'm going to talk to you about plankton tonight, specifically zooplankton, or predominantly zooplankton, and part of my PhD work on zooplankton in the Amazon River plume. So I have been studying for now a little over 5 years zooplankton off the coast of South America, and I'm gonna get into that and show you explain some of these pretty animals, or I think they're gorgeous, especially in the tropics. You think about really colorful flowers and plants. Well, the same goes for a lot of the marine species. You can think about it as they're really colorful, and that's like sunblock for you. So I'll show you some animals and kind of explain that in a little bit. But to start with, you guys have been looking around. So who knows about plankton? First off, who's this guy? Plankton, right? From what? SpongeBob, right? It makes my job a lot easier. All right. So this is plankton, and specifically, he's a zooplankton, right? So he's got one eye. So a lot of the animals I study are called copepods, and most people don't know what it is except for this guy. So that one eye, that's actually what a lot of them look like. They don't have a big smiley mouth like that, but that's kind of what they look like. So, but plankton is a generic term, and who read some of these signs and can tell me what it means? And the two different types of plankton. Anyone? All right. Okay, zooplankton and phytoplankton is what she said. So what's the difference? Any idea?
Zooplankton is like animals and phytoplankton are plants.
All right, so zooplankton are like the animals and phytoplankton are like the plants. Right, so that's it's kind of the idea that we have when you look, and if you learn about food chains or food webs, you think about primary producers, and that's grass or plants. And then you have consumers, and that's kind of you have caterpillars, and then a bird, and then a fox. So in the ocean, it's the same structure. You have plants, but everything's microscopic, or most of the producers are microscopic. So I'm going to show you a really quick video, and if you're really interested, and I think everyone should go and check this website out. It's called planktonchronicles.org. It's an institution over in Europe that have done a lot of really spectacular visualization of plankton, because these are microscopic organisms, or most of them are microscopic. So while you're out swimming in the bay, you're actually swimming with millions of these things. You just can't see them. So if you can check it out, there's a lot of just really good, and this book, they now have a book, which there might be a copy of here, but it's really good for visualizing it. So I have a video that's kind of a good primer overview that shows these animals while they're alive, and has a little bit of explanation about the different categories of plankton. So only a couple minutes.
Plankton comes from the Greek word planktos, which means wandering. Any living creature carried along by ocean currents is classified as plankton. It ranges in size from the tiniest virus to siphonophores, the longest animals in the world, and also includes microscopic algae, krill, or fish larvae. Some plankton, like these salps, drift all their lives. Others, like mollusks and fish, are only planktonic during their embryonic or larval state. When they reach adulthood, they settle or swim freely. Planktonic organisms play important roles in human life. Many microscopic species get their energy from photosynthesis. They absorb carbon dioxide and produce oxygen. Thus, they constantly renew the air we breathe. Plankton has also been a great provider of fossil energy. When it dies, it sinks to the seabed. This layer of sediment has fossilized for more than 1 billion years, producing our precious oil. Finally, plankton nourishes us. It's the basis of the food chain in which the large eat the small. Without plankton, there would be no fish.
Everyone's convinced, right? Plankton are great. All right, so again, I'm going to kind of really get into the differences here. And what the video mentioned there was, so we talked about zoo and phyto, right? But what about plankton? So the really important definition, and it kind of gets gray for some things like jellyfish back there, plankton comes from the Greek word for drifter. So depending on the currents, these animals, you think about, you go out and you swim, you move around, they're really small, so the physics is a lot different for them. It's kind of like them being, or you being in a big vat of honey. So if you're in honey, it would be really hard, kind of viscous, really sticky for you to swim through, and you would get pushed wherever that honey went, and that's because of the size. So phytoplankton, those plants, they tend to be in the surface waters because they need light. So photosynthesis, right? That's what plants do to use carbon dioxide and gives us oxygen that we breathe, but that's their energy. And then it goes down the food web. So again, similar to plants on land, but they're the producers, and there's a whole different a bunch of varieties. So these are some diatoms. They can be in the water column or actually down in, some of them are in the sediments. We call that benthic. And then you have these. This is if you have an electron microscope, so it's really, really high resolution, and this is called a coccolithophore. There are pictures of those, and these are really important in the North Atlantic. There are blooms of these. You guys have read about algal blooms. In the bay, we have some harmful algal blooms. These can be seen from space, so they get very, very large. Not the individual organisms, they're microscopic, but it's on the scale of hundreds to thousands of miles. So again, these are really, really important to oxygen production around the world. And then now we get into the zooplankton, which is what I like to study. Almost everything in the marine environment except for mammals, so all your whales and some of your birds that are marine, well, birds are not mammals, but your marine organisms that live in the ocean goes through a planktonic stage. So all the crabs, shrimps, lobsters, fish all start off as life in the plankton. There's some things like this copepod that lives their entire life in the plankton. And then there are other things. So this is a crab zoea is what we call it. It's a larval stage of crab. And you'll see they have really interesting spines. Part of that is so they don't get eaten. So again, you're in this environment where you can't really swim that well. A big fish comes along, wants to eat you. Well, a lot of fish, they just swim and eat with their mouth open. So if that spine is bigger than the mouth, they don't get eaten. So it's kind of a defense mechanism. And it also helps them keep afloat. So surface area really helps you when you're, think about when you're in the pool and you have, if you're trying to swim, you get a noodle, gives you a little more surface area, and then you float better. So that's kind of the same idea. One of those big pool noodles I like to hit people with. But, so some of the other organisms, copepods are one of the most abundant. So all of these organisms are called copepods. They're a type of crustacean. And I'll pass this around if you guys want to look at it. And you'll see these are all microscope images, because if you look here, a lot of these look like jelly beans. They're about that shape, but you can't really distinguish any of that in this jar. So I'll pass those around. And these are the most abundant organisms, well, multicellular organisms. There's lots of bacteria and viruses, but animals, we'll say, in the ocean. So these guys are really, really abundant. All right? Pass that around. And then there's a whole variety of other crustaceans. You have your, this is called an isopod. Maybe some of you guys have seen the videos or images of deep sea isopods. Some of these organisms in the deep sea can be the size of like a football. So that's kind of alien looking. These are called amphipods. These are some of my favorite organisms. And I have another example here of one of those that I'll show you in a second. And then we have ostracods and some of these, these are crab zoea again, so different life stages of crustaceans. And then jellyfish. We had the jellyfish back there. There's a lot of variety of specifically jellyfish, or we also call, there's scientists like to distinguish things by characteristics. So there's a lot of things that we lump into gelatinous zooplankton that are not specifically jellyfish, but that's kind of being nitpicky. And you saw this in the video. So this is called a salp. And I'll hold this for you and can look at it later. These are from Antarctica, so my lab does work in Antarctica too. And what's really, oh, you want to see? I'm gonna. He's gonna hold it. There you go. I'll hold it. This is a big jar. I don't want you to drop it. What's the yellow stuff? So we got a good question here. What are these yellow blobs? What do you think? Anyone have a guess? Well, it's part of a plant. It's part of zooplankton. Not seeds. It's actually their gut. So, all right, so we were talking about food. So zooplankton eat a lot of phytoplankton. The way this organism works is it's basically a barrel, and as it can kind of propel itself, it uses a siphon motion where it sucks water in, and every time it moves, it's eating. So think about that. Every time, if you were moving, you're taking a bite, right? So it's in, and it's filtering it. Basically, its gut is this net, and so it sucks water in and filters that really, really small phytoplankton we're talking about. So they're really important for a zooplankton ecologist because we study a lot of, we call it carbon cycling, but we study poop a lot, actually. So these things, they can make really fast, we call them fecal pellets. It's a little bit nicer term. But they can repackage that phytoplankton and make it a really fast sinking fecal pellet. And that's really important to kind of carbon cycling in the ocean, where you're taking carbon out of the atmosphere into the phytoplankton through photosynthesis, getting kind of eaten, and then repackaged and sinking to depth. So we study that a lot in my lab, actually. All right. You got skipped? Did you see it? There you go. Those are the copepods. All right, so this is on to my specific PhD work. This is the coast of South America, down here in the bottom part, and this is the mouth of the Amazon River bloom, or the Amazon River. So here you can kind of see all this sediment coming out. And so the Amazon is responsible for 20% of all fresh water coming from rivers. So 20% of all rivers that empty into the ocean, all rivers, comes just from that one spot. And so what that means is you have this really big pulse of fresh water bringing some nutrients and different kind of chemistry, and it allows for this gigantic phytoplankton bloom. So this bloom here, you see the bar on it says 1,000 kilometers, or about 600 miles. So it's actually about 621 miles. To put that in perspective, we have phytoplankton blooms. We just had a nice one, well, a big one, maybe not nice is not the term to use, that you might remember from last year in Virginia Beach, that we had all the bioluminescence. Think about the size of the Chesapeake Bay. So if that bloom was to happen in the Chesapeake Bay, it would be the entire bay, right? So these are massive scale phytoplankton blooms that are happening in the open ocean. So kind of like I was talking about in the North Atlantic, you have these blooms on a much, much larger scale than some of the things we see in the coastal environment. So again, when you're interested in tracking nutrients, carbon and nitrogen, it's massive scales that we're talking about here. So, and in the Amazon, there's kind of this really neat dynamics of a symbiotic organism there where it's making some nutrients available to kind of seed these blooms. So, and again, to show you again just the scale, so the Amazon River Basin is basically half of a continent draining to one point, and then the watershed is 3 million square miles. So that's a massive watershed. And that bloom, so here you can see this is actually the plume. Seasonally, this water will go all the way across to Africa. So you can actually track the chemistry of this water with satellites all the way to Africa. So this is a very massive kind of pulse of water. You had a question?
What are the local bioluminescent plankton that we see?
Local here?
Yes.
There are several different species of the phytoplankton. The major one that we've had recently is called Alexandrium monolatum. There are jellyfish that have some bioluminescence, and I believe that's a ctenophore called Nemiopsis is the bioluminescent one in the bay. Yeah, those are the two major ones that we'll see around here. One is the ctenophore Nemiopsis is zooplankton, and the algal bloom is called dinoflagellate. That's the Alexandrium. That's a phytoplankton. All right, so again, really just trying to drive home that this is a massive scale pulse of freshwater, and that really affects the physics of seawater. And so I'm going to try a demonstration in a minute that I don't know that it's actually going to work, but to Who has any idea about density? Does you know what it means when all right, let's see. Can you explain density to everybody?
Not really.
Not really. All right. Can you explain density?
Like how wet it is.
Yeah. What do I mean by saying something is dense? Let me look. So I'll use an example here. We have this styrofoam and we have this clay. Which one is more dense? The clay is more dense. So what density means is the amount of mass per volume. So if you think about kind of take a brick that's an actual brick versus a brick of styrofoam, right? One might actually weigh 5 pounds where one weighs less than a pound. So that's density. And when we talk about it in seawater, density is controlled by a lot of different things, but one of the big ones is salinity. So the actual amount of salt that's in the water affects density. So fresh water doesn't have salt in it, right? So it's less dense. And you can think about this as the mouth of the river over here, and then we're going out into the open ocean. And so the dark blue is the open ocean, and then this layer on top is the Amazon River. And what we study is kind of along this gradient, this change of low salinity out into a high salinity environment, what's happening with all the phytoplankton and zooplankton there. So that's kind of the entire part of my PhD in a nutshell. And I'm going to try this real quick to try and demonstrate this. It didn't work that well earlier, so I'll try. So what we have here is this blue. Yeah, you might want to stand up on this one. So we have here, this blue water is nice salty ocean water. And then what I have in here is clear, fresh water. This is the Amazon River, and this is the Atlantic Ocean. I'm going to try to highlight what happens when you have a density gradient and you get stratification. This may or may not work. Oh, watch out. I don't want to drip it on you. Is it stratifying? I don't think it is. Yeah, it didn't work earlier either. The water is actually too blue. I put too much food coloring in it, so then you couldn't. What's supposed to happen is maybe it'll settle out. I put too much blue in it earlier, and so when I tried to mix, what you see is basically what's on the picture up here. Is that salty dark blue water, and then that fresh water will actually layer. And so what that does is, because remember, plankton can't swim. We'll see if it settles out, but it probably just mixed. I tried to tweak it too with temperature, but that didn't work. So there you go. Best laid plans, right? They don't always work. Where'd my clicker go? Yeah. Thank you. Outstanding. I promise that's actually true. Don't go overboard with food coloring. That's the lesson. All right, so you guys can sit back down. That didn't work as well. But again, so stratification is really important. So you have this layer of water, and we have this in the Chesapeake Bay. You learn about estuaries and brackish water. That's the mixing of fresh and salt water, and you'll get layers. You'll have a salt wedge estuary. Some people call it. That's when you have a lot of fresh water mixing in. And so when we go out there and we sample in the ocean, in order to do this, we catch zooplankton with nets. And if you really want to look at that stratification, you have to use a certain type of net that we call a mock nest. And what it is actually 10 nets on one frame. And so when we put it in the water, when we come back up, normally with a normal net, if you just dip it in, you just take a profile. It looks like a triangle, and you're sampling the entire time. So what we do here is, you can see on the right image, see those bars across it. As we put this net in the water, we sit at a computer, and we know where it is in the water column, and we know the physical properties of the water. So as it gets to certain depth layers, we hit a button, and it drops this bar down and opens a second net. So when it comes back on board, each one of those nets has animals only from certain layers of water. So again, with that stratification, we're interested in how is the Amazon actually controlling where these plankton go. And that allows us to really sample very precisely in the surface layer of just the Amazon water, and then right below it, and then kind of the open ocean environment even below that. And so this is kind of the opposite. This is just a standard net where it's only one net on a frame. And let's see. So again, to drive this home, I don't know if you can, actually, can we hit the lights again just so we can see this picture really well? All right, this is actually a frontal region in the ocean. And if you look on the left side of the screen, it's like nice bright blue. And on the right side, it's a lot darker. And you can kind of see where all the white caps are on the left side and on the right side. So this is actually a very strong physical gradient where we had open ocean on the left. So this is salinity, like if you're at Virginia Beach. And then on the right, this is like salinity of being in the York River. So that you actually sometimes get that distinct of a line where you can sample on one side and be completely in the open ocean. And on the right side, it'd be like sampling in an estuary. So you're kind of in that coastal, in this top layer, and then as you punch through, then it's actually open ocean. It's really neat because here in those frontal zones, you get a lot of mixing. So here we saw a lot of sea turtles. We saw a lot of big marlin. We saw mahi, so if you're into fishing, because of all that mixing, there's a lot of the food is getting churned up, and so there's a lot of animals that like to find these areas, and they aggregate, and they'll eat a lot there. So this was really neat to see, and we sampled on either side of this, kind of towing through it alongside for a few days, because we only saw this happen twice in 3 years, so it was pretty neat to actually see it. So that's good. All right, and then to kind of real quick just show you some of the data that actually supports what I'm talking about. This animal here on the right, you see it's called Luciferfax sone. It's a decapod shrimp. It's a type of shrimp. It's not very big like the ones that we eat, but it's really important. This is a coastal organism though. So you find this all along the coast of North and South America. You can find it down in the Gulf of Mexico. And this graph, what it's showing is the abundance, so how many animals, and then along the salinity gradient. So as you go left to right, it's getting saltier. So you have this really high abundance in low salinity water, and then as you go out into the open ocean. But what's neat is that you can actually get that layered. So even though you might have this higher salinity water deeper and out in the open ocean, you'll still have these constrained in the upper layer of water. So I'll show you that real quick. So that's that's what this graph is showing. So now, left to right is abundance, and up and down is like you're going deep into the water. And you can see there's a lot of animals. That's what these bars mean. So as the bigger the bar, the more animals there are. They're really layered, constrained in that top 10 meters. So that's about 30 feet of water. It's the only place you found these animals, and that was at this high plume station. So what's happening is you have these coastal organisms basically get stuck in this low salinity plume water and get flushed out into the open ocean. So there's different ideas. Some things probably just get sucked out, and their life cycles, they can't live in the open ocean once they kind of settle out. These organisms, that shrimp that I was showing you, their life cycle is only about 35 days. So there's a lot of food in that water, so they can actually go through several life stages and reproduce multiple times in this very nutrient phytoplankton-rich water. So that's some of the things that we're seeing. So with that, I'll open it up to questions. We can do a little more show and tell with these animals. I've got some more that I didn't show you. But yeah, that's about the time. Any questions up here? You guys understand it all? You're pros? I got a thumbs up? No. No? All right. Anyone? Any questions in the back? Sarah has a question.
I'd like you to tell them how you became working with plankton. Like, what got you interested in that particular field?
Okay, so I actually, before coming to VIMS, I'm from Virginia. I went to UVA, and I did a lot of Chesapeake Bay work when I was at UVA. And for about a year, almost a year, I worked up at Oceanographic Institute on Cape Cod, studying harmful algal blooms. So in the Gulf of Maine, there are these massive, a similar phytoplankton bloom to what we have here, but it actually produces a very bad toxin. So it's a different type of Alexandrium. So I did that for almost a year, and I did a lot of this microscope work, counting and doing all these things. But phytoplankton, as important as they are, they mostly look a lot like circles. So doing microscope work with these really very diverse, but a lot of really kind of small things that don't look like animals wasn't as exciting to me. I liked the understanding these harmful algal blooms and what impacts they have, but I had an opportunity to come here on this Amazon project and really look at the response of the next step up in the food chain, the zooplankton, to and also a massive phytoplankton bloom. That one's not a toxic phytoplankton bloom, but it's still these same dynamics of having a really big phytoplankton bloom and what's happening and how is that affecting the food chain. And so that was kind of, I did that work for a year. I loved going out on boats. I went on a couple oceanographic cruises up in New England. So go out on a boat for, those cruises were 2 weeks. For all these projects, we were out on a boat for 5 to 6 weeks. So if you think about that, not getting off a boat for 5 weeks, even though it's a boat that's 300 feet long, think about not being able to move more than 300 feet for 5 weeks. Hopefully you like everyone. Most of the time, everyone gets along very well on these cruises, but yeah, so.
Were those NOAA ships?
These were not NOAA ships. There's a equipment sharing. It's through the National Science Foundation, and it's called UNOLS, is the equipment sharing. So a lot of institutes have a UNOLS vessel. So Woods Hole up in New England, University of Delaware, a bunch of these marine science institutes have these vessels that are actually owned by the National Science Foundation / the Navy, but operated by a research institute. So similar to how NOAA does those, but these are usually maintained by a university. So yeah. So we were on 2 boats out of Woods Hole and then 1 boat out of Scripps Institute of Oceanography, which is in San Diego. It all just depends on scheduling because it's all the same system. You just kind of depends on where the boat happens to be in to get it. So. Yeah, actually, I do. Well, more variety. I had one picture. Let me show you. So I talked about, well, let me turn around a second. So I talked about amphipods. These are probably my most favorite, just because they're, and specifically this type is called a hyperiid amphipod. And this entire part here, even though that dark spot there, that's actually all its eye. So basically their entire head is an eye. And they're very carnivorous, and they have really crazy body structures, and some of them have big claws. So they're really, really neat to look at. And I actually have an example of one here somewhere, this one. So these organisms, right, I called salps. These are the gelatinous ones. So there's a type of amphipod called Phronema that actually will catch this organism, keep it alive, but eat most of the internal parts of it and use it as a house and lay its eggs and then develop its young within the salp. So this jar has one inside of a salp, and there's a bunch of little dots that are the young. And so amphipods are my favorite. And this is a good example of one because they're really, really wild and have different, very unique life cycles. But there are a lot of other, there's some really neat, cool copepods. The pigmentation I was talking about, you'll see some. When we poured, here you guys can take a look at that. When we're in these open ocean tropical stations, you get this net on board and then you pour it into a bucket to actually do all the processing, so we then split it and preserve them in a preservative or freeze them for different studies we do. But when you pour it, a lot of times it'd be purple or orange or pink actually kind of coming out, and that's because when you put these in there, I think the very last picture had a bright blue copepod. You have these oranges and pinks and purples, and that's basically their sunscreen because there's so much. The water's so clear in the open ocean. Light penetrates really deep, and part of their UV protection is actually to develop this pigmentation to absorb those really. So here you go. You see this one, this bright blue, that's a copepod, and these were really, really interesting. And you can see that dark spot right at the top, like I was talking about with Plankton from SpongeBob? That's its eye. So, so there's some really neat copepods too. I like a lot of them, but if I have to pick amphipods. So. Any other questions? Oh, question?
How many different varieties of phytoplankton
How many different varieties? Well, if you want to talk to species level, at least hundreds of thousands, if not, yeah, at least in the hundreds of thousands. I don't know the actual number of species, but it's a lot of species of phytoplankton. Yeah, and zooplankton is very similar too. We're still discovering new species of both, so yeah. Good question. I don't know that anyone knows that exact number. It's always changing. It's always growing. We're always finding new species, so. Any other questions? Sorry the physics didn't work. I promise that actually does work.
All right, well, thank you, Brandon.
All right.